This video covers false prediction number 9. As usual, the link to the paper covering all of the false predictions is below in the comments section. Now, false prediction number 9 is initially pretty straightforward, but by the time we're done, it gets very interesting. Let's have a look. Evolutionists build evolutionary trees known as phylogenies. Evolutionists use a lot of data that show the similarity between the species. Species that are similar are closer on the evolutionary tree. There wasn't so much data in Darwin's day, and the evolutionary trees were simpler. After Darwin, one of the new types of data came from blood tests or serological tests. As Tim Berra explains here, these serological tests have to do with the immune system and proteins that are in the blood. So what is the prediction? These serological tests provided data around the turn of the century, a little more than 100 years ago, that were consistent with the old evolutionary trees. In other words, the new data confirmed the old data. Evolutionists found this to be powerful confirmation of evolution. In 1923, evolutionist H.H. Lane wrote that these data were most convincing of the evolutionary hypothesis. This view continued on later into the century. In the 1960s, Dodson wrote that the serological data were a most impressive evidence for evolution. Skipping down, thus, the results of serological tests support the theories of relationship which were originally based on comparative morphology. In other words, the new data confirm the old data. Continuing, they indicate that the serum proteins show varying degrees of homology, just as do gross structures. Okay, well, that doesn't make any sense because homology either is or it isn't. It doesn't come in varying degrees. Dodson means to say similarity, not homology. That's a mistake. But the point is, again, the new data confirm the old data. Continuing, quote, that this should be just a coincidence is not imaginable, yet it is exactly what would be expected on the basis of Darwin's theory. So there you have it. It is exactly what would be expected on the basis of Darwin's theory, that similar species have been formed by descent with modification from a common ancestor. So there's your prediction, a prediction of evolution. Continuing, quote, in general then, serology has verified the taxonomy which was worked out on the other bases, quote, end quote. Again, the new data confirm the old data. Back to Tim Berra now. He also cited this as strong evidence for evolution. So the prediction is that serological tests reveal evolutionary relationships. That's the prediction, and it is all false. Here is a quote from a leading evolutionist, J.B.S. Haldane, in 1949. Now, this is cited in a 1999 paper that I'm showing here. Quote, now, every species of mammal and bird so far investigated has shown quite a surprising biochemical diversity by serological tests. The antigens concerned seem to be proteins to which polysaccharides are attached, end quote. So it's falsified, and we'll see a lot more of this in a minute. So it turns out that the serological tests did not align with common descent. Instead, they contradicted the evolutionary model. And the key molecules involved are proteins to which a polysaccharide or glycan molecule is attached. These are referred to as glycoproteins. As we shall see, the glycoproteins, which are at the heart of the serological tests, do not fall into the common descent pattern. They contradict it. First, some terminology. Glycoproteins are proteins with a sugar molecule attached. Glycans are those sugar molecules. Glycans are found in other configurations as well. Sugars constitute an important part of biology, and its study is referred to as glycobiology and glycomics. So what we're going to see is that glycan synthesis is very complicated. Glycan structures are very complicated, and glycans strongly contradict evolution in several different ways. Okay, so back to that article. There is also no clear explanation for the extreme complexity and diversity of glycans. Skipping down, it is difficult to see clear trends or patterns consistent with different evolutionary lineages. So the serological test data contradict evolution because, for one reason, the glycans contradict evolution. They do not fall along the supposed evolutionary lineages. Continuing, there is not even an obvious relationship of structural diversification to the phylogenetic relationships amongst the species studied. And then parenthetical, the glycans on the mouse protein are more similar to those of the human than of those of the rat. So it doesn't fit the evolutionary pattern. And, quote, diversity in glycosylation is found at every level of biological organization, between species, within populations of the same species, and also among different molecules and cell types within the same organism. Glycobiology presents us with a world of incredible diversity. Diversity, diversity, diversity. Here's another article. Many glycans show remarkably discontinuous distribution across evolutionary lineages. Again, they contradict evolution, and glycans are very important. Okay, so glycans and glycobiology in general present several significant problems for evolutionary theory, much more than merely this false prediction number 9. In other words, this false prediction opens the door to a whole world of contradictions. Evolutionists should never have triumphantly claimed the serology tests as such a powerful confirmation because, in fact, it reveals exactly the opposite. To begin with, glycans are not template encoded as are proteins. Okay, so what does that mean? Glycan synthesis is not template driven. Genes do not encode them directly. Rather, glycans are assembled by enzymes. Glycans contain a variety of monosaccharide units and linkages, joining them in structures that can be branched and further chemically modified. You see diagrams like this, illustrating the assembly line. At each step, there is an enzyme doing its job. This assembly line starts in the endoplasmic reticulum, or ER, and then moves to the Golgi structure. The ER constructs a precursor structure, and the Golgi customizes the structure depending on current needs. Look at all the steps, and this is just a high-level summary. Here is another such diagram. You can see up there in the ER, the glycan is constructed as it is attached to an unfolded protein that is being synthesized. So you've got the ribosome pumping away, adding amino acids one at a time to a growing chain, and simultaneously you have this assembly line of enzymes constructing the attached glycan. This is incredibly complicated. Now here is a diagram that focuses in on the ER and its assembly of the precursor structure. Along the bottom you can see it starts off with a single phosphate group, and then there is a series of steps. In this first step, the enzyme is DPAG-T1. Let's zoom in on that. So DPAG-T1 adds another phosphate group and this other box called N-acetylglucosamine, which is based on glucose and looks like this. In the lower right, there is a kind of an acetyl group except with a nitrogen thrown in. This is a key building block in glycans. Here's a cartoon of the DPAG-T1 structure and how it sits in the ER membrane. It has 409 amino acids, so this is not a small protein. Now there is the active site where the lone phosphate group, which is not shown, is brought into proximity with the activated N-acetylglucosamine. Activated because a uridine diphosphate, that is UDP, is attached to it. Notice how the protein is folded up and different loops, which are far apart in the primary structure, come together in this tertiary structure at the active site. Here is a paper on the DPAG-T1 structure. You can see the active site depicted in the upper right. You can see the amino acid asparagine at the 185th amino acid in the DPAG-T1 sequence comes together just right with the 252nd amino acid, which is aspartic acid, and the 122nd amino acid, which is tryptophan, and the 301st amino acid, which is arginine. So, 4 amino acids, otherwise distant in the primary structure, all working together in precise geometry at the active site to add the phosphate group and the N-acetylglucosamine to the growing glycan precursor. All of that is happening just right. And if it isn't just right, and if you have mutations, for example, you get disorders and disease. Here's another look at how the DPAG-T1 structure sits in the ER membrane shown in the upper left here. You can see the structure in the protein databank also, and you can visualize the structure in different ways and rotate it and zoom in and out and so forth. And here's another look at the active site. I like the lower illustration showing the single phosphate group in green as the starting point and the N-acetylglucosamine with the uridine diphosphate attached, and how these entities coordinate with the active site amino acids just right. So that gives you some detail and idea of just one of the steps in the ER assembly line. It also gives you a better understanding of how glycans are not template encoded as proteins are. In other words, you don't have a gene that codes directly for the glycan. Instead, you have a gene that codes for the protein DPAG-T1, which in turn performs a task on the glycan assembly line, a very different concept, as this paper explains. Unlike the genome, exome, or proteome, the glycome is produced in a non-templated manner and is intricately controlled at multiple levels in the ER and Golgi apparatus. This assembly line is, quote, intricately controlled at multiple levels. The regulation and control are incredible here. You see, the glycan synthesis is not only controlled by DPAG-T1 and other enzymes, it is controlled by the environment and by the physiological conditions and so forth. There is a lot going on not shown in these diagrams, a lot of regulation and control. As this paper describes, at the evolutionary level, glycosylation provides eukaryotes with a rich combinatorial system generating an incredible diversity of glycan structures without prior genome modification. Skipping down, regulation of the glycoproteome, therefore, is essential for numerous biological and physiological functions and hence represents an unprecedented level of complexity. An unprecedented level of complexity, which stems in part from the fact that glycans are not template encoded. Quote, an important aspect of this regulation is that there is no gene encoding glycan structures per se, but 2% of the mammalian genome is estimated to encode genes involved in the synthesis of these glycan structures. 2% of the genome is involved in glycan synthesis. That's huge, and it means there is a tremendous amount of regulation. Skipping down, as noted, a prodigious enzymatic arsenal secures the biosynthesis of glycan structures. Indeed. Expression of these actors is a key parameter in the observed glycan structural diversity, and that requires a lot of regulation. So this isn't merely evolving a gene to get a glycan. It's evolving a large chunk of the genome to get that regulation. Okay, but that's only the beginning. We looked at DPAG-T1, but that was one of a great many other protein enzymes needed in the glycan assembly line. Glycan synthesis requires multiple protein enzymes. So with protein coding, again, you have a single gene coding for a single protein. Now, it's, of course, as always, more complicated than that. In fact, a single gene can code for multiple proteins, but that's another story. For now, the point is, when you have a single protein, it's generally going to be coded for by a single gene. That, in and of itself, is a monumental problem for evolutionary theory. I recommend you go look at false prediction number 5 for that. So I'm by no means suggesting that that's easy or simple, but let's move on to glycans by comparison. In eukaryotes, protein glycosylation involves hundreds of different molecular actors, including enzymes involved in the biosynthesis and transport of nucleotide sugars, as well as glycosyl transferases and glycosyl hydrolases called glycoenzymes. In humans, for example, more than 500 glycoenzymes take part in glycan processing. So that diagram we looked at was good, but it was just the tip of the iceberg. So for glycan coding, as opposed to the protein coding we just looked at, you have many genes, many proteins building a glycan. In other words, one gene codes for a protein that does one job on the assembly line. Another gene codes for a different protein, which does a different job on the assembly line. Another gene codes for another protein, which does another job, and another gene, another protein, and so forth and so on. You get the idea. A lot of genes, many proteins, one glycan. Glycans may experience some mutational constraints differently than naked proteins because they result from coordinated assembly pathways. Glycans are made by a network of enzymes, each building on or degrading the product of other steps, of prior steps. It would be an understatement to point out that this is beyond the reach of random mutations. Okay, next contradiction here. The glycan structural space is astronomical. Looking at this paper, most glycans are found on the outermost surfaces of cellular and secreted macromolecules and are remarkably diverse. A vast number of naturally occurring sugars can be combined to create a variety of unique glycan structures on lipid and protein molecules that modulate their function. Multiple enzymatic site preferences, as well as the use of stereochemical alpha or beta conjugations, create further diversity in where and how these sugars are linked to each other. In fact, altogether, these features imply the potential existence of about 10 to the 12th different branched glycan structures. In other words, a million different glycan structures. So a huge structural space that glycans present. Next, glycans are crucial to biology. This is not something you can go without. Glycans have a regulatory capacity and structural diversity beyond any other class of biological molecule. Glycans are tasked with such an amazing diversity of functions that no study of cellular interaction can begin without considering them. Very important. We propose that glycans predominate the cell surface because their physical and chemical properties allow the rapid innovation required of molecules on the front lines of evolutionary conflict. Okay, I hope a red flag went up for you there. I hope you saw the teleological language. In other words, we're having a reason for their design. We propose that glycans predominate on the cell surface because their physical and chemical properties allow. So teleological language, it's hard to describe biology, even for evolutionists, without using teleological language. We've seen that over and over. Continuing, glycan functions include physical and structural roles, adding rigidity or charge, extracellular matrix formation and morphogenesis, protein folding, transcriptional regulation, and information exchange between cells. Some glycan types occur in most organisms, and others are restricted to certain lineages. They are crucial to biology. Glycans are crucial to biology. Now here's another interesting paper. Sugars are the most functionally and structurally diverse molecules in the biological world. Glycan structures range from tiny single monosaccharide units to giant chains thousands of units long. Some glycans are branched. There are monosaccharides linked together in many different combinations and orientations. Some exist as solitary molecules. Others are conjugated to proteins and lipids and alter their collective functional properties. In addition to structural and storage roles, glycan molecules participate in and actively regulate physiological and developmental processes. Glycans also mediate cellular interactions within and between individuals. Glycans are crucial to biology. Okay, and finally, we come back to, yes, our false prediction number 9, the fifth of our contradictions to evolutionary theory. Glycans don't fit the common descent pattern that we pointed out earlier. We've already seen some quotes on this. You can find a lot more. This paper says that glycans occur in a discontinuous and puzzling distribution across evolutionary lineages. Examples of discontinuously distributed glycans are presented in Table 1. Glycans do not fit the evolutionary pattern. This paper looks at sialic acid. Sialic acid is a 9-carbon sugar which is often tacked onto the end of glycans in human biology, human glycobiology. and it's very unique to humans. So sialic acid biology is a study of human uniqueness. Quoting from the paper here, it seems unlikely that all these events occurred by chance. Thus, for want of a better word, we have suggested that human evolution was associated with a, quote, sialoquake, involving a series of related events in this system. So just make up a word and a process. The bottom line is they are observing very unique glycobiology in humans. Part of that involves sialic acid and how it's tacked on to the end of glycans. Skipping down here, the focus of this review is on the expected high frequency of differences in SIA biology between humans and our closest evolutionary cousins, the so-called great apes. The following points suggest that the human situation is unusual, and I won't read through these, but you get the point. Human glycobiology does not fit the common descent pattern. It's very different. Here's another paper. The extensive diversity observed for most mature glycan structures, summarized in Figure 2, indicates that glycan processing steps in the Golgi apparatus involve a large repertoire of organism-specific glycosidases and glycosyltransferases, as well as other enzymes required for specific sugar substitution. Organism-specific, that's the key. That's not common descent. That's the antithesis of common descent, organism-specific. So we have a lot of contradictions here. Not template encoded. Synthesis requires multiple enzymes, astronomical structural space, crucial to biology, and yes, our original false prediction, they don't fit the common descent pattern. So we've seen a prediction and we've seen the falsification, and a powerful one at that. What is the outcome of all this? This false prediction about blood immunity and serological tests exposes a much broader failure of evolutionary theory in the areas of glycobiology and glycomics, as evidenced in the literature, but how do evolutionists handle this obvious failure? Well, I want to return to this paper because this paper does show a lot of major important problems with evolutionary theory presented by glycobiology. It just doesn't fit the theory. It contradicts the theory. So what are they going to do about this as they're going through all these hits, all these major problems with evolutionary theory? And let's read here. Here we discuss the significance of this remarkable diversity, mindful of the oft-repeated adage of Dobzhansky that, quote, nothing in biology makes sense except in the light of evolution. Now, this is a rallying cry for evolutionists. Dobzhansky was a leading evolutionist, and he wrote this paper years ago with the title, Nothing in Biology Makes Sense Except in the Light of Evolution. Now, as usual, the paper was full of theological claims of why God would not have created this world, wouldn't have created these species. They must have evolved. It was, as usual, the theology did the heavy lifting. But in any case, this phrase is now a rallying cry for evolutionists. So you got to love this, that here are these evolutionists writing this paper about glycobiology, and they've got all these problems, these devastating problems for evolutionary theory, and it's just kind of like the one thing they can hang on to. Don't worry, we're gonna keep this in mind as we go through this. So this is what's happening. This is the outcome. Their evolutionists are not acknowledging the science. They're fighting the science. In spite of the obvious failures, evolutionists persist in force-fitting the glycomics data into the evolutionary common descent model. So I want to show you as another example of what the outcome is just the table of contents of a popular undergraduate level text, Molecular Biology of the Cell. Very popular text. This is the 4th edition that we're looking at. And I'm just going to do a little word study on the table of contents. So in this table of contents, DNA appears 130 times, RNA 76 times, protein 266 times, carbohydrate 0, sugar 3, glycan 7. So this is pointed out in some of these papers. I didn't read from all the quotes in those papers, but they pointed out that glycobiology just hasn't risen to the level of reputation and visibility that the genome and proteins get. And after all, those are core to evolutionary theory. So evolutionists really focus on genes because that's where the mutations are and the proteins that they create. And glycobiology just is kind of, in the background in these texts. And yeah, it's for structural purposes and a few other things. Now you might say, well, that's the 4th edition. That's way back there. They've changed. They've gotten better. So here's the 7th edition. Now this is the condensed table of contents. So the numbers are smaller, but you see the same sort of pattern. DNA shows up 13 times, RNA 6, protein 14, carbohydrate, sugar, and glycan 0 for all of them. So you can see the bias here. Glycobiology and its devastating impact on evolutionary theory is not really reckoned with in the literature. So you have a prediction, you have its falsification, and you have a response. It isn't pretty. So what do we say about the failed theory virtues in this case? And we've been looking at these theory virtues and how the false predictions bear on them. What are these theory virtues? Evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. So this is a good way to judge a theory is how does it do on the basis of these theory virtues? And these false predictions we're looking at are a good way of analyzing this. And they kind of serve as a framework to ask the question, how does evolutionary theory perform on these virtues? And on this one, it's very difficult to find any positives. It clearly fails on all of these. So I give the check on every single one of these. It is a terrible false prediction for evolutionary theory. Religion drives science, and it matters.